Thank you for your introduction. So today I'll be talking about modeling cancer progression and prediction of tumor behavior based on some experimental results that we obtained from our colleagues. So what am I going to talk about? It's actually two parts of my presentation. First, the investigation of the influence of electromagnetic field on different cancer cell lines. Second is modeling cancer progression when drug therapy is applied using data that we had from a real clinical patient. And finally, I will give some future improvements of our model and conclusions. So about this first part, we had our colleagues from the biological department had 3 different types of cancer cell lines, and they seeded it in a well plate. So they observed the influence of electromagnetic field acting on them. So we had 3 types of cell lines, and we also had control cell lines and the cell lines that were under the influence of electromagnetic field, and they observed how the cells behave. So I'm not going to talk a lot about biology because that's not my area of expertise, just to explain the basic experimental setup. So what we obtained through this experiment and what was our input to the model was the images of cells 24 and 72 hours after the start of this electromagnetic field treatment. So these images look like this. And on the other side, we had numerical data about the number of viable cells after 24 hours and after 72 hours. And we had the number of control cells and number of cells that were left when the treatment was on. So we wanted to model that, but first we wanted to model the electrical response of biological cells in terms of, finite element, using a finite element method. So we have one. For start, we have one cell that we have an extracellular, intracellular, and membrane domain, where this membrane represents practically the interface between the intracellular and extracellular space. So we have some electric field acting on them, but this field is not going to be uniform, and that's obvious. We're going to have some isolines that are going to look like this. This is extracellular potential. And then we modeled it using finite element, and we obtained results like this. This is the extracellular potential, intracellular, and transmembrane. So it is obvious that some of this potential goes inside this membrane, although it's really small amount, but it does go inside. So it is obvious that this electric field will, on some way, have some influence on cells. Sorry. So we can compare these results that we obtained with the analytical expressions for a single cell model. And for specific points on this membrane, we can see that our results with dots are in good agreement with the analytical results that were obtained, that were found in literature. And now we use the same thing with several cells, in this case 4 computer generated cells. And again, we had electric field, and again we have isolines that are, and the electric field is completely perturbed, so it's natural. And again, we have the distributions of potentials, and we see that the distribution is not identical in all cells, so it means that the position of cells, their distribution is equally important. So what we did now, from these experimental images, for start, we created a single cumulative cell, meaning why we did is it's an approximation naturally, and it's not good enough, but for start we wanted to simplify the model as much as possible to see what we can do, and later we're going to introduce further improvements in our model. So this is an equation that simulates the change of cell concentration based on diffusion of cells and proliferation, and also we extended this model to include the effect of electromagnetic field. And now it is obvious that in this model we have these constant diffusion and proliferation rate that are unknown. I mean, you can't find data about in literature that is specific for different cancer cell lines. So we had to estimate these parameters using a fitting procedure, and we obtained pretty small squared errors, which means that this fitting is pretty good. And then what I wanted to do next, I mean, this is a model that is somehow, I mean, it's really simple. So I wanted to introduce some kind of proliferation rate change, because it's obvious that cells are not just dying out of nothing just because the electric field is acting on them. I mean, proliferation rate has to change due to electric field. So I decided let's try out, because we don't have actual experimental data about this yet, but we're planning on doing this with Professor Jonovan, so we discussed it a lot in the pauses. But we used 4 different functions: exponential, Hill, logarithmic, and linear function, to see which one fits best the model. So if you look here, we have with red dots, these are the experimental values that we obtain, and these are the simulations that we performed using different functions. And it is obvious that the best functions were exponential and Hill, which means that well, I concluded that if you can try to model the proliferation rate with some different functions. I will discuss it a little bit later. So what we can do and what we can obtain is, if we have experimental images, we can create a numerical model, and we can have some diagram that shows us the decay of number of cells over time with the electric field acting on them, that's the blue line, and without electric field acting on them, that's the red line. About the second part, we had a patient with liver metastatic disease, and we had CT scan images every, I don't know, about 60 days. From the initial of the treatment, every 6 days we had CT scan images, and then we could create finite element models from those images in 3D. And we can numerically model the lowerness, I mean, the decay of tumor. And finally, we can get the prediction of further tumor growth based on the values that we have from experiment or from clinical data, whatever. So the mathematical model that we use is practically the same, just that we have this the diffusion coefficient that is not constant in all 3D in 3 axes, and we have the effect of drug therapy. So what I wanted to implement here is this, the idea was to use the EGFR signaling pathway somehow, because for those who are not so involved in this, the EGFR is the growth receptor, the growth factor receptor that initiates the growth of tumor and on all cells. So if the tumor is getting enough blood oxygen supply and stuff like that, he can further grow. And when this receptor comes to the EGF growth factor, the cell gets the signaling that it should grow further and proliferate. But if you somehow try to stop the signaling pathway, or with the drugs or with any other kind of treatment, then these drugs or other types of molecules will bind with EGFR, so the pathway is going to be blocked. And if this pathway is blocked, then the tumor will not further grow and it will decay in size. So I found this paper in literature that was modeling the change of proliferation rate with respect to the drug concentration, in terms that when the drug concentration is increasing, the proliferation rate is changing using the Hill function. That's why I tried to use the Hill function with cancer cells in the first part, and I implemented this in my model. So if you look at a simulation of untreated tumor, it's the black line. The blue line represents the simulation of tumor growth if you have only the effect of drug therapy in general and not on the cancer or on the cellular level. And the red line represents the final simulation that I was able to obtain using data that I have. So the entire simulation process looks like this. If you have these specific moments in time where you have drug therapy applied, the proliferation rate is going to decay like this. I mean, it's going to be constant, but when you apply the drugs, there is going to decay. Exactly the same is happening with the volume. So if you have this blue line that represents experimental results, and the red line represents the numerical simulation. So you have this tumor volume that is decaying, and I think that it is pretty good agreement of data that we were able to obtain for a simple model like we have. So I would like to conclude my presentation and tell about further improvements. I mean, it's obvious that every model has its own drawbacks and advantages. I'm modest by nature, so I'm going to assume that my model has more advantages in this moment. But the drawbacks are, first thing is that we have accumulated cell model for the first part, and it's obviously not good enough. It's just an approximation, so we have to improve that definitely. Second thing is that we have to include some kind of effect of electromagnetic field on cellular level, although it's not really my part of the job. Somehow we have to do this with biologists, but anyway, it's a drawback. And finally, we definitely need more experimental data, much more experimental data, to have a model that is better than this current one. And advantages are that we have some kind of agreement of results, so I think that means that we're on a good way, that this model can further be improved and improved and improved, and we can get somewhere. On the other hand, we have parameter estimation included, which is good because that's not something you can find in literature. And finally, we used some, we modeled drug therapy, which is also a good thing because you can't, you have to have that in the model. So that would be it. Thank you for your patience.
Thank you very much. Still questions?
I have two questions. The first one, your cell model. You demonstrated a couple of single cells. It's very important to have them confluent also, because after that you should groups of cells and they're changing their behavior and biology if they are confluent or they are single cells. I mean, this is very important because also if you have a tumor, you have a sensitive of cells. You have many cells which are working all them together.
Of course.
This is the first one. The second, the liver model. The liver is a very specific organ because the liver is very well vascularized by sinusoids, by the classical liver angiogenic architecture. And what's happened, what is specific for the liver, is not that the blood vessels are inviting the tumor. The tumors are going between the blood vessels. It's called in the literature cooption of this type of blood. How to say? It's not angiogenesis because by definition, but the tumor cells are going inside and not as you demonstrated that the blood vessel coming to the tumor. This is a conceptual difference with what's happened in life, in the real life.
Okay.
I just had two questions. Maybe I didn't get it. When you talk about diffusion of what? I didn't.
Cells.
Diffusion of cells.
They are migrating in the domain. And diffusion, it's a reaction diffusion model, mathematical model, that uses diffusion of cells and proliferation. Because in literature, I found that, for example, glioblastoma has greater diffusion coefficient than proliferation, because the cells are expanding in the domain.
These cells are in the cell culture, and they are attached.
No, they are in patient, real data from patient. From CT.
No, but the first one was...
The first one is for cancer
cells,
cells, yes.
So they are in the culture.
Yes.
Because they are basically attached to the surface. So
Yes. Yeah, the COVID.
Yes, but when you, in the results that we obtained, the diffusion is really small. So it practically agrees with what you say.
yeah, maybe I wouldn't call it just diffusion, maybe migration or something.
Okay, it's a biological term. I said I'm not a biologist, so I don't, maybe I mix the terms. It's okay.
Just the second thing, which I probably missed, the patient, the liver patient was treated with the antagonist of the
Yes.
VEGFR?
VEGF inhibition, and I'm not sure of what were the drugs that were applied, but he had drug therapy applied, yes.
In that respect, there is a migration assay, exactly what she's saying. It's already established in the biology. They just use that one. It's a called migration assay.
Okay.
Yes, exactly. Coming back to the migration, if you use this, and I know that a lot of system biologists they use this diffusion reaction equation, right? But there your flow of cells is proportional to the gradient of the concentration, right? You have something times gradient of concentration. And that means physically that the individual object is doing random walk, right? That was the idea of Fick's law. So the molecules, they are just moving around in normal chemical species, and then you have this diffusion. And I'm also always wondering, what are the cells doing? Do they really random walk? Because you would say, okay, they are sitting there, they are very slowly, but they do random walk, and this is why they are spreading. Is this really the biological phenomenon, or do the mathematical biologists just, incorporate nice mathematical models?
They are in the cell culture, I mean, in the dish, they are attached to the surface.
But this would mean no diffusion.
They are moving. They are moving.
But why are they moving?
Because the cytoskeleton, they are moving, changing their interaction, actin filaments, and they are moving. Even they are a specific type of carcinoma. This is not Johnny Walker, but it's a walking carcinoma. It's called working.
Okay, so they have random walk.
Yeah,
They
yeah.
have random walk.
You
Because that is what
could
you
see that one. You could
They just
document
go
that.
around.
Yeah.
Free.
Okay, yes.
Can you go to the graph with the size and the time?
Then physically it's different. Migration.
Let's see, what one, which
The
one?
last one with the size and the time.
Okay, okay. This one.
This one, yes. Why is this effect that increases and decreases and then increases again over time?
Because this is the tumor volume. It naturally, the tumor is going to grow, but then the decrease is due to the drug therapy. I mean, the drugs are killing down some tumor cells.
And
So
then
the
they-
number of cells is decreasing, but then due to proliferation, they are rising again. The number is rising again.
Okay, thank you again very much. So, our three professors, they have to leave now to airport, so we want to say hello to, thanks again them for coming. And I hope we will continue our collaboration and Switzerland, first of all, the presentation and also with Scopes project and other projects. So thank you again for your time.